Our mouth is continuously exposed to oral microbes, and it is a balance between these microbes and our host tissues or soft tissues within the mouth which determine whether we have a healthy oral mouth or an oral disease. And so within our lab we're actually engineering human healthy soft tissues of the oral cavity, the mucosa, and we actually use these to look at and investigate host microbe interactions, that is interactions of our oral mucosa with potential pathogens, but also to look at how the abutments and the filling materials brought into the mouth also interact with these soft tissues, potentially causing adverse effects. In our lab, we are actually world leaders in making organotypic mucosa. Our organotypic model gave exactly the same results as the clinical study and the dog study, showing that it is a very valid alternative. And so now actually we're in the position that we can actually train our early career scientists to actually use these methods and hopefully in the future to replace all the use of living animals for this type of research.